So we get started. So wanted to bring everybody up to speed on the quiz on Thursday. I think I sent you an email the other day. And we indicated that that was going to be over. Let's see, section. It was just over section 8.3. Did everybody get that email? Yeah. Okay. So have you gotten into chapter 8? I would hope. And if so, how's it going?
It's difficult.
It is a challenging chapter. Yeah, you're right. Absolutely it is. Especially that section 8.3, I think, is probably one of the more challenging sections because it has a lot of the glycolysis, Krebs cycle, electron transport stuff in it, which is usually what students have the most difficult time with. I think that the first two sections, not so bad. It's kind of going over enzymes, what they are, how they work. A lot of that should be review or not too terribly difficult, although we can certainly talk about any of that too. So everything's on the table. So maybe I'll just open it up like I typically do and see where you're having issues, where you'd like to get some clarification on stuff or review. I will begin pulling up the PowerPoint for chapter 8. So tell me what's on your mind.
We have to know all of the steps for all of like glycolysis and the Krebs cycle.
Well, you don't need to know all the intricate individual reactions, of course. That would be absolutely crazy to try to understand and to learn. Even a graduate level course wouldn't probably make you do that. I think it's important to know what do you begin with, what do you end with, and what is made along the way. I think if you keep those things in mind, it becomes a little more palatable. So for example, if I asked you to describe what molecule or molecules does glycolysis begin with, what molecules does glycolysis end with in terms of what's produced at the end, and what does the cell make along the way from beginning to end of glycolysis, could you answer that? Okay, so what would the answer be? What would the molecule or molecules be that we start with in glycolysis?
Glucose.
Right, start with glucose. And as the name implies, glycolysis, glucose being broken apart. Yeah. And so what do we end with? What molecule or molecules?
Is it in the pyruvic acid stuff?
Yeah. There's two molecules. Important to put that coefficient in front of that word, though. 2-pyruvic acid. Another name for that is pyruvate. Sometimes you see that described in a video or in a book. Don't let that throw you. In fact, your lab book or lecture book, I'm just looking here at page 142. They're using the term pyruvate. Let's see, when they talk about actual glycolysis on page 244, here they're using pyruvic acid. Now, I think if I was the author of this book, I would try to be consistent, although they do say pyruvic acid and in parentheses pyruvate. So maybe they're trying to do like I'm trying to do, and that is, make you aware that there's more than one term that you could use. But either one is correct. So absolutely right. 2-pyruvic acid or pyruvate are made at the end of glycolysis. And then the third part of that is, what does the cell make between starting with glucose and ending with the 2-pyruvic acids? Yeah. Can you give me, anybody else give me one of the byproducts? Because Miss Bundig seems to know her stuff. That's great. Anybody? What's made in glycolysis other than pyruvic acid?
1 ATP.
Yep. At least one? Now, don't confuse this with citric acid cycle. I think there's more than one. How many ATPs are made in glycolysis altogether?
Is it 38 or is that the citric acid?
No, that's the whole process
Oh,
of the three: glycolysis, citric acid, and electron transport. We're talking just about glycolysis here. How many ATPs are made in glycolysis? If I asked you that question, how many are made?
2.
Be careful how you answer that. Stephanie, did you say 2?
Is it 2?
You're warm. The question was, how many ATPs are made? Answer is 4. If you said 2, what were you thinking about?
Isn't it that 2 ATP are used in the process?
Well, that's true, but what was Stephanie saying when she said 2? She was assuming what?
I just remember reading 2 ATP, to be honest.
Okay. There are 4 made, and as Samantha said, there are 2 degraded. So there's a net gain of 2 ATPs. Now, again, if you said 2 ATPs are made, I would assume you were doing the math. 4 minus 2, meaning there's a net gain of 2 ATPs in glycolysis. But if you look at that pathway on page 244, you will count 2 ATPs degraded initially in the process, and then 4 made toward the end, giving you a net gain of 2. Okay, anything else made in glycolysis? Net gain of 2 ATPs, 2 pyruvic acid, anything else? Answer, of course, is yes. What else is made in the middle of the process?
NADH.
NADH. How many of those? 2, right? What does NADH do? If you had to summarize it at dinner tonight, when you have dinner with your folks or your husband or your significant other, what does NADH do? If they ask you, you're going to answer what? You're going to sit there dumbfounded. I have no idea. No, you know, you've been studying this. You know what NADH is. What did we define it as or describe it as?
A bus.
Yes. A bus that is empty or full. NADH.
It would be full.
Right. It's full of passengers. The metaphor we gave was a school bus full of students. Only this is a molecule full of what? I know Monica knows. Go ahead, Monica.
It's full of electrons.
Right. It's carrying electrons. It's an electron carrier. That's what you got to remember. Electron carrier. Why do we want an electron carrier? Well, because the last part of aerobic and even anaerobic respiration is something called electron transport system. In order to transport things, you need electrons. And so those are gathered up by our NADH and FADH2 coenzymes is really what they are. They're coenzyme molecules. Okay. What happens to those two pyruvate molecules or pyruvic acid molecules if we are talking aerobic respiration? Where do those pyruvic acid molecules go? Now, if you are totally clueless, you need to become unclueless quickly. You should know this.
Do they lead into the Krebs cycle?
Exactly. That's our next stop along the way, right? Krebs cycle or citric acid cycle. This occurs where in a bacterium? And don't say mitochondria because they don't have them.
The cytoplasm.
Right, cytoplasm. That's where glycolysis took place. Yeah. And in the citric acid cycle or Krebs cycle, technically before we actually enter the cyclical series of reactions. So when I think of the citric acid cycle, I think of that in my mind's eye. I think of this circle of reactions. Maybe you do too. But before we can enter the circle, if you will, of reactions, there's something that the book describes as a linking reaction. When I teach this in A&P or biology, I came up with a name for it, and it's pretty close to what the book calls it. I call it the bridging reaction. That's just my own term that I come up with. But see, to me, when I say that or think that, that tells me it's bridging glycolysis and the citric acid cycle. Like the book calls it, it's linking the two. So it's the same idea, whether you want to call it the linking reaction or the bridging reaction. It's not really called that, but that's what it does. So in that linking or bridging reaction, each pyruvic acid gets converted into what's called acetyl coenzyme A. And that linking reaction alone produces several interesting, important compounds other than just acetyl coenzyme A. And if you look on the top of page 246, you'll see what I'm talking about. Just as we get below the pyruvic acid, in the first green part of that process, it's labeled A and with a yellow circle. There you see pyruvic acid converted to acetyl-CoA. Now, if you look at that acetyl coenzyme A, how many carbons does it have? They're pink C's. There's two of them. Now, each of the pyruvic acids have 3. So that other carbon, of course, goes into the production of carbon dioxide. Right? You see that on the left-hand side. The cell will also generate another NADH molecule. Remember, this is an electron carrier. Where is the source of those electrons that NAD plus gets, whether it's in glycolysis or here in the citric acid cycle or in the bridging reaction? What is the source of those electrons? We don't often talk about that. But I think we need to understand they don't come from outer space. They're coming from what? It's kind of like a real obvious thing if you just think about it. Go ahead.
I don't know. Is it the transport system? I don't know.
No, that's where they're going to go.
Okay, I was like, I wasn't sure.
Yeah. No, you're in the right track. That's where they're going to be taken. But where are we getting them from so that we can take them there? That's the question. Where are they coming from?
From the hydrogen.
Okay, true. And that hydrogen is part of what? It's part of the molecule that's being impacted by that reaction, right? So let me just pull up the citric acid cycle. You all see that? Okay. So here's that conversion of pyruvic acid into acetyl coenzyme A, and there's the CO2 that I was describing. Let me see if I can blow this up a little bit more. Okay. So here's NAD plus becoming NADH. It is being reduced. So what I want you to understand is that when this NAD plus gets electrons, and Jocelyn is right, they really come from the hydrogens that get kind of pulled off, but those hydrogens are coming from the pyruvic acid, right? They come from the molecule that's being influenced by this reaction. This is a single reaction catalyzed by an enzyme. And we could even go back to glycolysis here. If I can do that easily, I'm not sure. Here we go. The same thing applies here. We can zoom in on where NADH is being made here. Okay. These two NAD pluses that become reduced to NADH, they're getting their electrons from glyceraldehyde 3-phosphate. Again, do you need to know that name? No. It's not important you know the names. It's more important that you understand the basic concept. And it's hard. This stuff is not easy. I know it's not easy. It's very abstract. And it's challenging. I do understand this is not easy stuff. But it's not really super difficult if you just kind of try to get the main concepts. So back to what we were just saying, in the citric acid cycle, or I should say leading to it, back to our bridging reaction, we are producing a CO2 and an NADH. So here comes that 2-carbon acetyl group. Now we're really technically entering the formal Krebs cycle or citric acid cycle. And just as soon as this coenzyme A, it's called, the CoA is just short for coenzyme A, that was basically slapped onto this 2-carbon fragment of the pyruvic acid. I can't tell you why, but just as soon as it gets added on, it gets pulled off. And that's what this is called. This is showing this arrow coming back. That leaves us, just imagine this being gone. This leaves us with the 2-carbon compound, an acetyl group is what it's called. And that acetyl group combines with a 4-carbon molecule, whose name is not important, but if you want to look it up, there it is. And when you add a 4-carbon to a 2-carbon, you make a 6-carbon compound. And there it is, right there. It's called citrate. What's another name for that? Anybody know?
Is that citric acid?
Exactly. It's the same thing as citric acid, and that's where it gets the name citric acid cycle or Krebs cycle. Either term is correct. This is named after Hans Krebs, who was, I forget what nationality he was. I want to say Swedish. He won a Nobel Prize in chemistry for figuring this out back in like 1905 or something. But it's also called the citric acid cycle based upon the first compound made. Okay, so again, what do we make along the way? For every crank or revolution through the Krebs cycle, can you tell me what is made in a cell? Now, this could be in a prokaryotic cell, or this could be in a eukaryotic cell. This is happening in your cells right now. Do I need to know all these intermediary names and compounds and all that? No, you don't. But you need to know what's being spun off, right? So we have one ATP made directly, which you see at the bottom. And we've got 3 NADHs produced, not counting this one made in the bridging reaction. And then we also make an FADH2 molecule where FAD becomes reduced to become FADH2. And I likened the FADH2 in the Zoom lecture to a smaller school bus. In other words, it has the same role to play as the bigger NADH school bus. This is just a smaller, stubbier yellow bus. Doesn't carry quite as many students, but it still carries electrons, okay? So they're close cousins, these two, NADH and FADH2. They're close cousins. They're both coenzymes. They're both electron carriers. And I really like how they put this in here. This is really good because you have to think about, well, okay, we're making all these coenzymes. Why are we making them? Because as I said a moment ago, they're taking their passengers, the electrons, to this last mechanism called electron transport. Now, one could also argue there are some other compounds made here besides ATP and the 3 NADHs and the 1 FADH2. One could also talk about some waste products produced. Just like we produced the CO2 up here, notice that we make a couple down here as well, right? And that makes perfect sense because if you think about this first initial molecule made, citric acid, 6 carbon, and this 4 carbon compound made at the end of the preceding cycle that combines with the next 2 carbon to make a 6, right? This keeps on cranking through, assuming you've got inputs at the top. You got to account for the loss of 2 carbons, right? 4 minus 6 is 2, and there they are. Those are the CO2s. Now, if this is happening in your cells, which it is, okay, what are you exhaling right now? CO2. You all know that. You had no idea where it came from, probably. You just know you exhale CO2. You're taking oxygen. Everybody knows that. Well, now you know technically where that CO2 gets generated. The majority of it is generated here in the citric acid cycle, right? Yeah. Is there any made in glycolysis? No. It's all made, 2 of them are made in the citric acid cycle, 1 is made in the bridging reaction. That is where that CO2 you're exhaling right now, that's where that is coming from. It's coming from chemical reactions. Okay. So how do we feel about glycolysis and Krebs cycle?
I have a question.
Sure.
Like on an exam or quiz, if you were to ask how many NADHs were made in the Krebs cycle per pyruvic acid, we don't count the one that's made in the bridge reaction then? Or we do?
Technically, you would not. Technically, you would not. I'll just say technically you should not add that first NADH.
Okay.
Yeah. So the answer, of course, would be 3 for pyruvic acid. If I were to ask you how many NADHs are made in the Krebs cycle per glucose, the answer would be what?
6.
Right. If I said how many NADHs were made in the Krebs cycle if you started with 3 glucose molecules? Ooh, more higher level math here.
18.
Well, 3, 6 times 3, 18. Right? Because you got to remember, each glucose makes 2 pyruvic acids. Each pyruvic acid goes through the cycle once. Right. All right. Any other questions about glycolysis or Krebs cycle? Are we good with that? Are we still confused by that?
Can we go over the end products of the Krebs cycle again?
Yeah.
So I know it's 3 NADH. Is it just 1 ATP?
Yep. 1 ATP down here.
1 FADH?
2.
2, okay. And
It's no. I didn't say 2 FADH2. That's the name of the compound, FADH2.
oh, oopsies. Okay.
Okay. Don't mix them up. This chemistry is a little different than this one. It's got a subscript 2. So FADH2, there's 1 made per revolution. There's 3 NADHs made per revolution. And then of course the 1 NADH made up here in the bridging reaction. And again, if you want to talk waste products, you're also generating 2 CO2s down here, and you're generating 1 CO2 up here. We don't really talk too much about the CO2 because it doesn't really do much for us down the road, like the NADHs and the FADH2s and the electrons they're carrying. That plays a real important role in the next step, right? Electron transport. But I think it's worth noting that CO2 is a common byproduct of any sort of aerobic respiration, whether it's in the bacterium or in our cells. So that has to be given off and is as a waste product by cells. It would be, it would just diffuse across the bilayer and the cell wall in a bacterium, and then you and I would, of course, dissolve into the bloodstream, and the majority of it would be carried as bicarbonate in the plasma, AMP1 stuff. Right. All right. Let's see. Any other questions about that? Any questions on glycolysis or citric acid cycle? Are we good with that? Sort of, kind of. I would make a note card for each or something, and try to really spend some time examining those figures in the book. You know, this one. And this one. And don't get all caught up in, like, where is this 6-carbon compound broken and what are the names of these intermediary molecules? You don't need to know that. You do need to know glucose. You do need to know pyruvic acid or pyruvate. You do need to know how many NADHs are made. You should know how many net gain of ATP there are. And that's about it. All right. Anything else on those? Then let's talk about the electron transport system. Now, I don't know if you've had a chance to watch some of these videos, or you've watched the lecture, the Zoom lecture I made the other day that talks about this mechanism. I'm assuming you've all looked at it. If you haven't, you should have by now. And while it initially looks a little daunting to understand, it's really not that bad if you just take your time and study what's going on. Now, what the book does and what I did in the lecture is I started talking about this as it related to what occurs in a eukaryotic cell within the mitochondrion. And then later we transition into where this happens in the bacterium. So again, we can play this video if you want. You should have already watched it. Do you want to play it? Should I play it? I don't have a problem.
Sure.
You said go ahead? Okay. Let's see if this will work.
When glucose is oxidized during glycolysis and the Krebs cycle, the coenzymes NAD+ and FAD are reduced to NADH plus H+ and FADH2. In the mitochondria, the electrons from NADH plus H+ are transferred to the electron carrier proteins, and the protons are transferred across the membrane. As the electrons move from cytochrome to cytochrome down the electron transport chain, more protons are carried across the membrane. Cytochrome C transfers electrons to the cytochrome C oxidase complex. Protons are also transferred to the outside of the membrane by the cytochrome C oxidase complex. The cytochrome oxidase complex then transfers electrons from cytochrome C to oxygen, the terminal electron acceptor, and water is formed as the product. The transfer of protons generates a proton motive force across the membrane of the mitochondrion. Since membranes are impermeable to ions, the protons that re-enter the matrix pass through special proton channel proteins called ATP synthase. The energy derived from the movement of these protons is used to synthesize ATP from ADP and phosphate. Formation of ATP by this mechanism is referred to as oxidative phosphorylation.
So let's watch that one more time and we'll kind of pause as we go.
When glucose is oxidized during glycolysis and the Krebs cycle, the coenzymes NAD+ and FAD are reduced to NADH plus H plus and FADH2.
Oops. When we use the term reduced, does anybody remember what that means?
Is that to add like an electron? I could be wrong.
You're adding
When
what?
glucose
You're right. When a molecule is reduced, it gains what?
I want to say electrons, but I don't.
Yeah, right. Exactly. It gains electrons. That's exactly what happens. And so we have been gaining electrons in the formation of the NADH and FADH2 up to this point, haven't we? Those full school buses, they're loaded with electrons. Those are reduced school buses. Yeah. Now, when they get to their destination, which in this case is the inner mitochondrial membrane, they're going to open the doors of the school bus and the electrons are going to spill out. And those two molecules will then become what? What's the word? They were reduced.
Oxidized.
Oxidized, right. They get oxidized. And then when those empty oxidized school buses are done leaving their passengers off, guess where they go? Logic would tell you what? What do school buses do?
Go get more.
Back to
They shuttle students. That's all they do.
Start over.
Yeah, they're going to go where? They're going to travel back where? Where do you want them to go? Tell me where you want them to go. I'll take them there. Some are going to go where?
The Krebs cycle.
Some are going to the Krebs cycle. One's going to go where? Bridging reaction. Follow me? Where's some others going to go? Starts with a G.
glycolysis.
Right. Are you seeing the big picture here? Yeah. So try to make this a story. And if you do, it suddenly makes it easier to understand, right? So these NAD plus and FAD plus oxidized school buses are simply going to go where they need to go to find electrons. And then once they have electrons, they're going to all go to one place. And that, in this case, is to the inner mitochondrial membrane in a eukaryotic cell. Okay, are we good with that? Okay. So
is oxidized during glycolysis and the Krebs cycle, the coenzymes NAD+ and FAD are reduced to NADH plus H plus and FADH2. In the mitochondria, the electrons from NADH plus H plus are transferred to the electron carrier proteins, and the protons are transferred across the membrane. As the electrons move from cytochrome to cytochrome down the electron transport chain, more protons are carried across the membrane.
okay, so the NAD has already left. And here's the FAD that's just given off its electrons. It's going to leave, and as we said, go back to pick up more electrons. But where do the electrons go once they've gotten off the buses here? Well, if you've noticed here, Some of these, all these electrons have been passed from protein to protein to protein to protein to protein within this inner mitochondrial membrane. So if you follow the red E's, those are electrons. Some of them started here, if they came off the NADH. Others of them start their journey a little bit further down the line, and they're going to get passed through another similar, actually the same series of proteins from this point forward. As those electrons get passed from protein to protein to protein, what's happening across some of these other proteins? Well, you're seeing the hydrogen ions. They're passing out of the matrix into this intermembrane space. That's what these blue guys are. They're hydrogen ions. They're basically protons is what they are. And those protons get pulled off of the NADH and the FADH2. That's kind of sort of the source of the protons as well as the source of the electrons. So let's see what happens ultimately to these electrons. Do you know what the final electron acceptor is going to be? If it's aerobic respiration?
Is it oxygen?
It's oxygen, right? It's oxygen. And you'll see that form here in just a minute. And those electrons will combine with oxygen and hydrogen to make water. You'll see that. But notice that as these electrons are getting passed through these various other proteins, you see the pumping out of the hydrogen ions into this intermembrane space.
Cytochrome C transfers electrons to the cytochrome C oxidase complex. Protons are also transferred to the outside of the membrane by the cytochrome C oxidase complex. The cytochrome oxidase complex then transfers electrons from cytochrome C to oxygen, the terminal electron acceptor, and water is formed as the product.
So as we just said, water is the end product made as a result of oxygen accepting those electrons and then combining with some hydrogen to make H2O. Now, you always thought that your source of water came from the faucet, right? Or from liquids. And you're right, it does. But guess what? Your cells make it too. Yeah. But you didn't know that. You don't get all your water from what you imbibe. Some of it's made. Yeah. Do you see how the hydrogen ions are accumulating outside in this space because of the flow of these electrons? That's what pushes them across.
The transfer of protons generates a proton motive force across the membrane of the mitochondrion. Since membranes are impermeable to ions, the protons that re-enter the matrix pass through special proton channel proteins called ATP synthase.
So notice these hydrogen ions, they can't get back in through any of these other proteins, embedded proteins. They pass through a special one, which happens to be an enzyme. And we know that because it ends in ASE. ATP synthase. It's aptly named because it's here that ATP is synthesized or made. So as these hydrogen ions move back into the core of the mitochondrion, called the matrix, that allows for the phosphorylation of ADP. And what do you make?
ATP.
ATP, right.
The energy derived from the movement of these protons is used to synthesize ATP from ADP and phosphate. Formation of ATP by this mechanism is referred to as oxidative phosphorylation.
Now, you remember back in glycolysis and also in the Krebs cycle, we made a couple ATPs as we spun off those processes. The way that ATP is made is a little different than the way the majority of ATP is made here. As the video said, this is called oxidative phosphorylation. It's formed as a result of the hydrogen ions moving through this ATP synthase molecule. Back in glycolysis and citric acid, it's made through something called substrate level phosphorylation. It's a different mechanism. It's still phosphorylation of ADP to make ATP, so that's the same, but the mechanism is different. So it's worth just making note that the majority of ATP produced by cells is through this mechanism called oxidative phosphorylation, again, a result of electron transport, but some can be made in a different way, not as many, but some through what's called substrate level phosphorylation. So I'll just open it up for questions. Is this making sense? Is it totally confusing? Is it clicking? What do you think? Somewhere in between. We can go to a very similar diagram in your book, which is this, and it's a little bit hard to see this, but it's based virtually on the same diagram that the video used, right? You should recognize a lot of this stuff. Again, the inner core of the mitochondria, the matrix, the inner mitochondrial membrane in blue, the outer mitochondrial membrane in pink, and the intermembrane space between the two. These are the embedded proteins in the inner mitochondrial membrane. Now, when you look at this as a whole, what does this remind you of? Phospholipid bilayer embedded proteins. That is what we defined as the what back in A&P 1 or Bio 1. If I told you phospholipid bilayer with embedded proteins, what should immediately come to mind? Immediately. What organelle of the cell? We can call it an organelle. It technically is. But
Cell membrane.
cell membrane, right. Exactly. Cell membrane, right? Remember that? Fluid mosaic model, it's called. Well, the fact of the matter is, in many eukaryotic organelles, we have a very similar architecture. Phospholipid bilayer with embedded proteins. Here you see it. You don't notice it here in the outer mitochondrial membrane, but I can guarantee you can see the phospholipids. But there's embedded proteins here too. This is a common theme, structural theme throughout cells, eukaryotic cells. It's not just the cell membrane, it's many membranous organelles too. Okay, so all this is saying is here we have NADH and FADH2 giving off their passengers in the form of electrons. There you see it, follow the black arrows. These are the oxidized empty school buses, right? Okay, we're good with that. Yes? Very good with what's going on so far. They've just acted to shuttle electrons here. And again, here they tell you where they came from. So good job, textbook. Right. The electrons are getting passed from protein to protein to protein to protein to protein. Ultimately, they will be accepted by oxygen, called molecular oxygen, and some hydrogen ions to make good old H2O. As a result of these electrons getting passed from protein to protein to protein to protein, eventually making their way here, what is moving into the intermembrane space, i.e. pink arrows? Hydrogen ions. So my question to you is, if we did not have electron transport take place, would the cell be able to make ATP via oxidative phosphorylation? Yes or no? If we didn't have the electron transport system functioning, would we have ATP being made? The answer is a definite no. Why not? Because it's the shuttling of the electrons that is the force that pushes these hydrogen ions out here. And then they come back in through here. And that's where we make the ATP. So electron transport it does lots of things. It helps to make water at the end, but more importantly, it provides the force, it's called the proton motive force, that pushes these protons out into the intramembrane space, where they then diffuse back into the matrix via the ATP synthase, and that allows for the oxidative phosphorylation process to occur, that being phosphorylation of ADP into ATP. Again, make a story, think about a process. Suddenly, this doesn't become like rocket science. I mean, it's not easy, but it's not like impossible to understand if you just think about what's going on. Right? So for each NADH that we started with here that dumped off its passengers, the cell makes approximately between 2.5 to 2 3/4 ATPs. And for each FADH2 that dumps its electrons off, the cell makes about 1 3/4 ATPs, approximately. Why do I bring that up? Well, because if I want to know how much ATP a cell makes aerobically, I need to know a couple things. I need to know the total number of NADHs and FADH2s made, and I need to know what yield comes from each of those to come up with a final number. You following me? And I do that on one of these slides. Where is it? This slide. That's what this slide talks about. This slide says that in aerobic respiration, for each glucose that we start with, that goes through glycolysis, Krebs cycle, and electron transport, the cell will generate either a bacterial cell, well, yeah, we'll just say a cell for now. You're generating somewhere in the neighborhood of about 30 ATPs per glucose. And the reason that it's not exactly 38, which is assuming a 3-to-1 ratio for the NADH and a 2-to-1 ratio for the FADH2, is because, as I say here, there's some active transport costs here, and it's not exactly a 3-to-1 or a 2-to-1 ratio. It's a little bit less than that. So if you know approximately 30 ATPs per glucose, that's great. Now, the other thing that I failed to mention is, where does this happen in a prokaryotic cell? And I hope you know the answer. If you've watched the PowerPoint, you should know. Because prokaryotes don't have mitochondria. You all know that. If you don't know that, you're in big trouble. we said this years ago, right? Months ago. Prokaryotes lack organelles that are found in eukaryotic cells. Other than what organelle, maybe you could argue?
Ribosomes.
Right, ribosomes. Okay, so here's the answer to the question. Where does electron transport take place in a bacterial cell that lacks mitochondria? It's here in the cell membrane. That's where those embedded proteins are, including cytochrome C, where that molecular oxygen combines with the electrons and the hydrogens to make water. And then here's our big funny looking what? ATP synthase, right? Where the ATP is generated. And instead of having an intramembrane space like we would have back in the mitochondria, of course, we're talking about a periplasmic space. So it's the very same process. It involves virtually the very same proteins. It's just a different location, right? Yeah. And I guess the other thing that I would point out, if I can find this slide that I'm envisioning in my mind. Sorry about all this bouncing around, but here, this one. Here's what we just spent the last 40 minutes talking about, right here, right? That's this. In anaerobic respiration, glycolysis occurs, citric acid cycle occurs, or Krebs cycle, electron transport occurs. But instead of having oxygen as a final electron acceptor in the production of water, in an anaerobe, it's other compounds that ultimately acquire those electrons at the very end. And things like hydrogen sulfide and nitrates and nitrites are produced. Different chemicals are produced by anaerobic bacteria. With the ATP numbers, again, it says here varies with microbe. That's all I'm going to basically say about that. We can't easily tally it up like we kind of did with aerobic respiration. And in fermentation, notice we have glycolysis occurring like we did in these other two, types, but we don't have Krebs cycle, we don't have electron transport in fermentation. And we only generate 2 ATPs per original glucose. So it's a fairly inefficient process from an energetics point of view. It's important though, because some bacteria and yeast cells, which are eukaryotes, they rely on fermentation. That's how they make their living. That's all they can do is ferment. And that's not a bad thing. It just means they don't crank out as much ATP as a eukaryotic cell does. That's okay. They're happy yeast cells, you know. I'll stop here and just see if there are questions, things you'd like clarified. Is this making sense? I mean, I think we've basically gone over what the quiz is going to be over. We covered section 8.3, really. Again, don't get all hung up on the names of the chemicals, because the book does, it gets into some detail there, like the carriers within the electron transport system. They talk about on 247, the various complexes. They're just embedded proteins. That's all those things are. But you don't need to know the names. I guess I would know cytochrome C, and that simply is the last one of the last carrier molecules there, where oxygen, gathers those electrons at the end and combines with the hydrogen to make water. It's called cytochrome C. Well, I guess they do say here complex 4 catalyzes reactions, blah, 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 completing the electron transport process. Now, most of the videos just go to cytochrome C, so that's where I'm going to end it. Questions? Are you feeling comfortable with it?
I feel better about it now.
Okay, good. How about other topics related to chapter 8 that you'd like to ask about? Enzyme action. How enzymes work, what the energy of activation is. Our goal is to, get through this chapter, I would say, somewhere around now, Wednesday or so, because the second half of this week is devoted to the next chapter, which is 9. And so I will be posting probably Thursday, the first of, I'm not sure how many lectures. I really never know how many they're gonna take. Just in kind of perusing through this, I'm gonna say probably 3. Yeah, probably 3 lectures for 9. Anything you'd like to review? Okay, I will stay in this Zoom room for another 18 minutes or so before the second micro lab starts. All right. So I guess I'll post the quiz like I've done through the Respondus system, right? And I kind of know some of you are starting a little bit later in the day, Thursday, so I have names and times, so I'm going to stick with those times. Samuel, tell me, are you normal time or are you starting at 6 p.m.? I forget, or is that just the test?
I can do normal time.
Okay. Now, what about subsequent exams? You don't know yet?
As of right now, I think I should be good.
Okay. You can just tell me.
I'll let you know if anything changes on that, obviously.
Yeah, great. Let's see, Sarah, you're doing 8 p.m., right? Liners?
Yeah, 8 p.m.
Yeah. Let's see who else is here. Ashley, are you doing 7 Thursday, right? Still doing 7?
On this Thursday, yeah. The next 2 Thursdays we have off from clinical, so I don't know if you want to change it or just leave it so there's not a bunch of back and forth.
Whatever works for you, Ashley. I'll leave it for now. If you would rather do it in the afternoon, you just tell me.
Okay, I'll email you just so it doesn't get all confusing and the back and forthness of it.
Okay, what I'm going to do is just operate on the assumption that Thursday at 7 is going to be good, and you tell me if you prefer to do it earlier. Not a problem to switch back, at some point.
Okay, well then, yeah, next, I don't know what the date is, sorry. Look at the calendar. Next Thursday, the 25th, I believe we're off, and the 1st.
What about those 2 dates?
I don't have clinical, so I could do the
Oh.
quiz. And I think the 1st is the exam.
Yes.
I could do those at regular times.
So you would prefer to do them at regular times?
Yes.
Okay. And this week you need to do 7.
Yes.
Okay. I'll make a note then. Anybody else have questions? You can bug out if you want.